We're just going to give it a minute for everyone to join. Okay, seems like we have a good amount of attendees. We can get started. Hello, everyone, and welcome to today's webinar on the topic of elevating your soil health expertise, utilizing BeCrop tools to enhance crop advising. My name is Sarah Basiri, and I am the Global Head of Marketing at Biome. And joining me today is our very own agronomist and product specialist, Gus. For today's webinar, we'll be providing an overview of Biome, details about BeCrop Advisory Program, and a deep dive into our BeCrop technology and updates to BeCrop 3.0. We have dedicated some time at the end of this webinar to answer all of your questions. So please don't wait until the end. Use the question panel on your screen and enter your questions as we go, and we'll get to them right at the end. If we run out of time, we'll be able to answer all your questions through email after the webinar. So make sure you answer all your questions or add all your questions. If you have any sort of suggestions for us for future webinars, if you just want to share your thoughts with us, add them into the question panel, and we'll definitely take all of those into consideration. Lastly, this webinar is being recorded, and a copy will be emailed to you within the next 24 hours. Now, before we get started, we were interested in seeing how many of you are currently BeCrop advisors or have enrolled into the program. So if you guys don't mind, we're just going to launch a quick poll and have you guys enter in your answers. Okay, I think we can close off the poll now. Great. Okay. Thank you all for contributing to the answers. And I'm going to quickly get started again to provide a quick overview about Biome and just a little overview about BeCrop Advisory Program. And then our colleague here, Gus, will do a deeper dive into BeCrop technology. Okay, a little bit about us. Biome was founded in 2015, and we have become one of the foremost global ag tech leaders and setting the standard for soil health with our BeCrop technology. Sorry, I'm just trying to get through the slides. Our BeCrop technology connects soil biology to agricultural decision-making to optimize farming practices and reverse the degradation of arable soils. We do this by obtaining soil microbiology insights that lead to better informed decision-making. We leverage the soil microbiome, environmental data, and machine learning to improve soil functionality and agricultural sustainability. Over the last 7 years, we have developed the largest database of taxonomic references, which is currently at 17 million and is continuing on growing. BeCrop technology has been utilized in 45 plus countries. We have analyzed over 170 crops. We have served over 2,000 customers. We have analyzed over 50,000 samples and have influenced over 500,000 hectares of land who are implementing biological soil analysis. We also have partnerships with top retailers and license our technology to labs like Water Ag and Region Ag Labs who are now providing biological soil sampling and analysis to their customers. How BeCrop technology helps the agriculture community. Under BeCrop technology, we offer 3 different types of solutions. And those are BeCrop tests used by farmers and advisors to provide functional soil analysis and translate complex data into user-friendly reports. We have BeCrop trials that are utilized by retailers and manufacturers to measure their agricultural input performances and provide actionable insights. And lastly, with our BeCrop rate, it's a metric for sustainability. It empowers the entire food chain system to track and promote regenerative practices. Of course, you can learn more about each of these solutions by visiting us on our website. Now, to tell you a little bit about our BeCrop advisory program. BeCrop is quickly setting the standard for soil health. Oh, I'm sorry, I don't know why I am now unable to share the right. Sorry guys, my computer is a little slow. Okay. So BeCrop technology, as I said, is setting the standard for soil health across the globe. And we wanted to provide access to this great technology. And we have been successful doing this by certifying passionate advisors and agronomists and forming partnerships with some of the world's best organizations who are helping us elevate this great initiative. And BeCrop Advisory is basically a free certification program for growers and agronomists who are looking to become leading experts in soil health. The education and certification is offered virtually and can be completed at your own time from anywhere in the world. And it's currently offered in English, Spanish, and French. And we're currently working on translating it in Portuguese as well. Today, we're proud to announce that just under 1 year, we have over 1,500 enrolled into the BeCrop advisory program with 28 graduates. And as I mentioned, this program is free, and with just a couple of hours of online training, one can become certified like our own very favorite AgTech leader, L. Toops, as you can see his picture on this slide. You can unlock many benefits of by becoming an advisor. So some of these benefits include obviously free certification and a soil health education access to free client management and project design tools that Gus will provide an overview on shortly. Free listing on our website as a BCA, where farmers can independently connect with you. Access to science-based recommendations that can help you optimize input costs and detect early disease risk. And the ultimate benefit of becoming an advisor is setting yourself apart from other industry players, connecting with farmers and providing the latest technology that's truly changing the world of agriculture. We also offer continuing education units to certified advisors, and some of the other benefits are exclusive discounts on BeCrop tests and unlocking benefits like features as being featured as a guest speaker on our webinars, podcasts, or becoming a thought leader on our ebooks and great type of material that we share with the industry. Becoming an extension of Biomaker's team by receiving merchandise, customized marketing materials, attending BCA closed-door webinars and events, and receiving quality leads and getting featured in our social media channels that have over 20,000 followers, and also connecting and collaborating with other advisors on our gated portals that are currently available to BCAs only. And the best of all, having access to our experienced agronomists like Gus, who can help you interpret test results and understand the health of your client's soil to provide better or the best services that the industry can possibly offer. We will be providing links on how to join the BCA program in the post-webinar follow-up email. We'll also be holding closed-door monthly webinars for BCAs and providing further training on BeCrop technology, how to decode the science to better understand agronomical decisions and advice, and do a deeper dive into reports. So be sure to register for the program and join the community. I'm going to stop sharing my screen now and pass it on to our very own advisor here, who is going to talk more about BeCrop technology and all the new updates that we have made. Gus is our very own agronomist and product specialist, and he has a background in agroecology and regenerative crop systems research. He received his graduate's degree from University of Florida in natural resource conservation before obtaining his master's degree in agriculture and environmental science at the University of Texas. Prior to joining Biomakers, Gus spent time working as a laboratory manager in University of Florida Horticulture Science Department and served as a product consultant for a biological ag product manufacturer. So he comes to you with lots and lots of experience from the industry. Gus, thank you so much for being here and joining us today. We're so excited to hear you and learn from you and hopefully be able to utilize BeCrop technology to its full potential. I will sign off now and be back at the end of this webinar to get to our Q&A portion of this webinar. Over to you, Gus.
Thank you, Sarah. Appreciate the introduction and excited to go over some of the updates and new features offered with BeCrop 3.0, the third iteration of BeCrop. And so I'm going to go ahead and share my screen and just share some of the examples of what we've changed and improved on. Sarah, are you able to see my screen currently?
Yes, I am, Gus.
Okay, I just want to make sure the slides are going out to the audience. So, here I'll be focusing on how and utilizing the BeCrop tools to enhance crop advising, and I'll try to leave maybe 15, 20 minutes or so for questions at the end. So I'll try to get through this fairly quickly and conclude with a quick overview of a BeCrop report in the new format, just to provide a little taste of what our reports will look like going forward. So the big update that folks will notice when entering our BeCrop portal are two new modules. So previously, when logging into the BeCrop portal, you'd go straight to your results overview and the dashboard that shows samples that have been received, along with tools and pages that can go over your results. We now have integrated two new modules, the planner, the project planner, as well as BeCrop Tracking, which we'll be talking about in more detail in a minute. So some of the values behind these improvements and changes we've made just include what we've always been doing at Biomakers, and really what we're looking to do is promote independence for agronomists, crop consultants, those in the BCA program, and continue to set the standard for soil health. We're constantly learning, collecting more data, and improving the science and our understanding of soil health. So in my opinion, this is essential to continuing to ensure that we don't get complacent with the technology and tools that we have, accepting that there can always be improvement and new angles, ways to look at things. And of course, as more research comes out, not just within Biomakers, but across the entire field of soil microbiology, it's very important for our team as we continue to integrate that new research into our technology to ensure that our technology is robust and accurate as possible. So we continue to develop intelligent computing systems that can answer the most comprehensive questions about soil health, and continue to give back to the community by empowering agricultural professionals with the science and tools they need. And we pride ourselves on creating trust and transparency, not just in BeCrop, but how we operate and how we apply our technology to the greater agricultural community. So with BeCrop 3.0, the main updates that we've integrated, again, focused on promoting independence, accuracy, transparency, and full control with our BCAs and agronomists. The BeCrop Project Planner, so that first module that we looked at when looking at the homepage for the portal, the Project Planner is a tool that allows you to create documents of proposals for trials and larger projects to provide clients with some idea of what the project will entail, and we'll talk more about that, the specifics that we've integrated there in a minute. Some new additions to the BeCrop portal also include the tracking module. So we now offer a module that second module that was added on to the portal allows projects and samples to be tracked throughout the analysis process to provide easily accessible gauge on where the samples are and how close they are to result delivery. And then we've also added some improvements to result delivery and analysis. This includes updates to the BeCrop computing and data systems, so the science stuff behind the microbial functions that we look at in our BeCrop reports in order to make it more robust and accurate, and then some changes to our BeCrop-based products, to our report interfaces, which again, we'll go over just a brief overview of a new version of a report at the end here. So first, we have, again, the new module interface, where in addition to the results module, we have the tracking and planner portal. And we'll start by discussing the project planner. So again, the project planner is designed to allow BCAs to create projects, to create proposals for projects within minutes. And this is really designed to be very automated, very self-explanatory, and allow for both independence and to save time on the end of BCAs who are looking to get projects set up and get experimental treatments designed. So you can create a proposal with this project planner within minutes, independently create these without needing assistance from biomakers, and allows you to tailor the project and the proposal document to the needs of the business, the farm, whatever client you're working with. You can build a proposal on here with or without agronomic expertise on the actual plot of land that you're developing this project for, thanks to standardized project design. So number of samples and factors like that are automatically integrated into this project planner so that you don't need to be certain about how many samples are needed. We provide a minimum in order to answer research questions, and this allows for more transparent and control on really what the projects look like, as well as maximizing the robustness of the results. We also offer full control and transparency on pricing so that you can integrate any additional pricing for your service fee and interpretation fee to ensure that you can be completely transparent with the client on what the final price will be for the project, not just the price of the BeCrop tests or trial. You can manage all of your proposals under one platform, so they're all saved in one place, and we'll talk more about that in a minute. And one of the real benefits of creating a proposal, a document that lays out each project, is that it allows you to take into account the problems or questions, research questions that the client is interested in during the proposal process, so that you can revisit this after the results are delivered. And this really also helps set consistent expectations with the client on the type of results and reports they will receive. In fact, the project planner documents, the proposals that are generated, include examples and listing of the specific results and reports, so that you can give a client an idea of what sort of documents they'll expect to receive with the results when those results come in. So within the project planner, we have we document and provide a compilation of all proposals that you've created. So once proposals are accepted through the project planner, they'll be entered into the BeCrop portal so that you can start to track and monitor the results. This makes it much easier to keep track of all the projects you have ongoing with various clients. Allows you to revisit and then look into the tracking portal. Full control and transparency on design of all projects, so you can revisit and look at changes that might need to be made. And again, managing all these projects under one platform just makes it a lot easier to keep track of, as well as straightforward display of trial design and setup on that proposal itself. So each proposal comes along with a simple diagram breaking down the trial design, if it's a trial looking at treated and untreated, if it's a BeCrop test project, looking at informing management practices, yield or diseases. It allows you to look at what specific parcels or zones or blocks that will be sampled within the project. So overall, just keeping everything in one place and making it easier to keep track of projects as they proceed. Next, the BeCrop Tracking. So again, this interface, which we have an example of here, places all projects in one location with project titles and dates, looking at the different type of the project, as well as an update on how far along each project is. Here's another example of what this tracking module looks like. And really the benefit here is to facilitate, to ensure that any information that is needed on your end or from the client is included so that we can get those results fast-tracked and facilitated for delivery as soon as possible. So what this includes is a GIS map as well as calculation of where to collect samples. We provide with each step in which we need some action to proceed with sample delivery. We have a check mark or symbols showing that we need that step to be addressed. So from sample collection to shipping, notification of sample being received at the lab, as well as the need for entering specific metadata that we may need for trial analysis, all the way to results. So these are all steps in the pipeline of completing a trial or project from sample collection to delivery. So this is much easier now to keep track of, to ensure that you're aware of how far along the project is, and so that you can set realistic expectations with your client. Now going into our updates to the BeCrop reports and our analysis. And this is personally what's really exciting to me, because I think that it's very important to continue to develop and improve technology, especially in a field as novel as soil biology. There's a lot out there that the field of soil microbiology in general continues to figure out and yield new findings on. And at Biomakers, we're committed to continue to integrate that into our technology and continue to improve the accuracy and robustness. So within our results module, the module that many of you may already be familiar with if you've used the BeCrop portal, we've offered some updates to improve the visualization and accuracy of our results. In this case, this is our new visualization tool, which allows parcels or blocks to be viewed side by side and look at highlights as far as the soil microbiome functionality. So this visual analysis, as you can see here, allows to look at plots individually or by comparison. You can filter results, compare and contrast, different areas, the levels of soil microbial functioning, and it just makes it easier to understand the scores and metrics that we're looking at here. Much better, in terms of client facing, oftentimes much better to get a visualization of their fields to deliver to them. So this is just a quick rundown on what this interface generally looks like. So we have a highlight of problematic soil microbial function indexes, so indexes that are on the lower range that can be improved to improve overall soil health. And then we have beneficial or metrics that are not problematic, that are within ranges that are generally indicative of high soil health and soil microbial functionality. So a bit of a deeper dive and simple to view highlight of the problematic and high functioning soil microbial indexes in each field. And now I'll just go through a couple quick case studies on this results module. So in this case, this is a BeCrop yield project, and the problem is that a grower is struggling with yield limitations in a field where they have dedicated major investments in their fertility input program for several years. So this is a situation that I'm sure many agronomists and crop advisors are very familiar with, especially with the costs of fertilizer in recent years as growers continue to ramp up their fertility program and invest more in an effort to improve their yield. Oftentimes they can run into bottlenecks in which they have issues with plateauing or declines in yield. So in this case, a grower wants to figure out what's standing in the way of their yield, how can they improve it? And the solution is to collect several BeCrop tests within the low yield field to determine the biological bottlenecks that are limiting yield here. And in this case, across this field where they took several samples, we can see the nitrogen pathway and overall biocontrol rates here are in the lower ranges, are in the problematic area. So these are nitrogen, of course, one of the most essential nutrients to plants and can definitely stand in the way of yield for many of our major agronomic crops. And then biocontrol, the microbes that naturally suppress pests and pathogens, specifically the four we look at being fungal pathogens, bacterial pathogens, nematodes, and insects with life cycles in the soil. Here on our 0 to 100 BeCrop score, that biocontrol rating stood at 39. So there is potential that those pests or pathogens could be problematic and present an issue to yield as well. So those are two areas that these results would indicate that they can potentially improve upon. And we can also see here the metrics that are high, that are within the ranges on our BeCrop score, our 0 to 100 score, which again we see the legend for down here. Phosphorus and potassium are within the upper end of that medium range, so some room for improvement. But what this would tell us in terms of yield, phosphorus and potassium are less likely to be a bottleneck or a hurdle than nitrogen, biocontrol, and some of these ratings that are in the lower ranges. So this can ultimately help you give the client a very clear visualization on how they can potentially improve their yield and inform their agronomic decision-making on management practices or biological ag inputs that can hone in and serve the right purposes to improve their yield. In this next case study, this is looking at a BeCrop management project. So in this case, a grower wants to compare fields with different management programs to determine which field shows superior soil health and nutrient cycling. So the solution here would be to collect several BeCrop tests in both fields and compare the results to see which field is showing higher levels of soil microbial functionality. And across field A and field B, here we have the comparison on the left, the visualization of each field on the right. And field B is showing much higher functioning phosphorus and potassium pathways, so higher levels of microbes known to help unlock and make P and K plant available, improved carbon pathways. So here we just really have the highlights, but when you're online in this interface, you can scroll through all of these differences. And rather than showing the overall BeCrop score on the 0 to 100 index, when we run a comparison, what we're seeing here is the difference in BeCrop score, which ones were higher in field A by this score, and which ones were higher in field B based on this score. We can see much higher levels overall in potassium and phosphorus in field B. And if you scroll down, we'd also see the carbon pathways functioning higher, as well as higher overall functionality. So the conclusion here would be that field B, the management practices conducted there showing improvements overall in soil health and microbial functionality. So this, again, very easy way to show a client a visualization and provide them with highlights of the strengths differences between their fields to answer their questions in terms of how their management practices are influencing soil health. So in addition to those changes to the portal in terms of visualization and data interpretation, we've also made some really exciting improvements to our pipeline for calculating the data in our reports. And this is just a quick rundown on some of those changes we've made. Our soil quality index continues to be improved and made more robust. We've launched a more accurate index. And the soil quality index, as many of you know, is designed to show basically how the impacts of management practices are impacting microbiome ecology and soil health. So if there's any metric on the BeCrop report that is a very synthesized metric, a standalone metric that provides a rundown on how management practices are positively or negatively impacting the soil microbiome, our soil quality index is really our standout metric there. And we continue to improve it by integrating more data and expanding our database. We've also improved on and created a new metric for resistance. We're transitioning the resistance metric to resilience, which is an improved metric for network calculations on both the bacterial and fungal populations. Very similar to resistance, resilience will be intended to look at the ability of the microbiome to remain resilient to stress and disturbance. So any sort of disturbance event that can negatively influence the microbiome, that can cause a major shift in population, This is resilience, just like resistance previously, but more robustly looks at that entire microbiome of how effectively it can respond to stress and disturbance, to things like tillage, fungicides, drought, biotic and abiotic disturbances alike. This looks at how effectively that microbiome can withstand those disturbances. We've also continued to update and change our computing libraries, so updated scientific references to deliver more accurate results. As I mentioned, in a field like microbiology that's changing very fast, new species being discovered, new functionalities and ecological synergies being discovered in the wider scientific literature. Very important that we continue to broadly expand and use the scientific findings out there that have been established to improve our results and not become complacent with our technology. And similarly, we've continued to update our taxonomic database. So as our updated taxonomic biodiversity indicators involve improved precision based on more taxa detected. So more species, more taxa that we've detected within BeCrop tests, and more references identified within our database. As Sarah mentioned, we've continued to expand the number of references. This allows us to account for more microbes and better understand how those microbes interact and influence soil health. So the value proposition here is improved power, precision, confidence, and robustness in our analysis. So you can be confident that we continue to improve on our results and ensure that we're maximizing the accuracy of the agronomic insights we can provide. So we have updated our BeCrop format. Now, aside from, of course, the changes potentially in the soil quality metric and then our new resilience metric, which will be replacing resistance, most of the report other than that is very, very similar. And we'll do a quick rundown. Just to go over a quick BeCrop report example, I'm just going to do a quick time check so that we have enough time for questions. But I'll give a quick rundown from top to bottom of the report for those who may be less familiar with our BeCrop reports. And then I'll also hone in on some of those areas we've changed or improved upon. But in general, for the most part, our reports provide pretty much the same information that they did previously. You'll just notice some different formatting and colors. So the header, everything's pretty much the same here on our new format, including information to provide location and parcel, the crop type. You can input other metadata like soil type and variety, as well as the date the sample was pulled. The genomic or the population focused data on the top, the numbers and ratios we look at are the same. We still provide the total number of different microbial species identified, which in this sample, hopefully that's able to be seen on your screens, but 583 different microbial species identified here. The total load of bacteria and fungi on the left provided in units per gram is the same, as well as the arbuscular to ectomycorrhizal ratio, looking at the ratio between the two main types of mycorrhizae, the symbiotic types of fungi that support that form mutually beneficial relationships with plants and support plant growth by essentially trading resources with plants. And then the fungi to bacteria ratio, also the same, a general indicator of soil health that over time with regenerative ag practices and soil health focused practices, we aim to shift closer to a 1-to-1 balanced ratio. The summary report here, we see some differences from our previous reports. Soil quality now appears very clearly as a standalone indicator, which it is. While it is related to biodiversity functionality and resilience, our new indicator, soil quality itself stands alone and is calculated independently. And we can see our new format allows it to look like a standalone indicator. You'll notice resilience replaced our resistance rating here on the right, but measures very, very similar phenomenon, but but looks deeper and is more robust than the resistance metric. Going down to soil quality, we see here displayed very similarly, but again, soil quality is shown as a standalone independent metric. Going on to the second page here in plant health, this is of course where we look at diseases and pathogens. We provide a risk level for any detected pathogen, and this is specific to crop, as I'm sure many of you know. So depending on the crop type selected, that will decide what major diseases and pathogens we look at and what risk level they present. The risk level provided is based, of course, on the presence and abundance of the pathogen, as well as natural biocontrol agents that can suppress pests and pathogens. And those biocontrol agents are also shown down here. Again, we look at fungicide, bactericide, insecticide, and nematicide agents. So these are bacteria and fungi that are capable of suppressing these types of pests and pathogens, and very important to informing the crop protection program of growers, and allowing them to determine how effectively their soil is naturally suppressing fungal pathogens, nematodes, insects, and bacterial pathogens, so that they can potentially provide or move towards independence from fungicides, fumigants, and other crop protection products, which can occasionally pose a risk and negative effects on soil health. In this particular report, we can see these biocontrol levels are lower, so this might be an indication that a biofungicide or bioinsecticide could be beneficial if they're looking to continue to improve the natural suppression of these pests and pathogens. Now moving on to the moving on to the next couple pages, but it looks like my PowerPoint might have frozen. There we go. All right, so page 3, the next part of the plant health section. This also remains unchanged from our previous format of the BeCrop report, and this is where we look at phytohormones and stress adaptation metabolites. Now, remember, we're not measuring the levels of these specific phytohormones in the soil or in the plant. We're measuring the levels of microbes that are known to produce these phytohormones, and in the second section, stress adaptation metabolites. So auxin, cytokinin, and gibberellin, of course, considered plant growth regulators, PGRs. And the way I like to think about this when talking to growers or agricultural professionals, of course, in agronomy, it's well established that plants need to produce these phytohormones and some other metabolites to support their growth and metabolism. But what's less commonly considered in agronomy is that the microbes in the soil can produce these PGRs as well, and essentially donate them to the plants to help improve the plant's growth and yield. So this is essentially looking at the natural ability of the microbiome to provide PGRs. And as many of us know who have applied PGRs foliarly, they can have a huge impact on growth and yield of a crop. This next section, stress adaptation metabolites, is where we're looking at both several phytohormones. So on the right, ACC deaminase, salicylic, and abscisic acid are all phytohormones. But what's different between these phytohormones and the 3 above, auxin, cytokinin, and gibberellin, is that these 3 here are involved in plant stress response. So helping a plant deal with forms of stress like drought, pathogen, helping prime a plant's response to pathogens, salinity stress. So these are all phytohormones that are involved in that ability of a plant to adapt to stress. And then on the left, we have several metabolites that microbes produce and that can help them deal with things like heavy metal stress, salinity, lack of iron availability in the case of Siduriphore. So these are all compounds that microbes are involved in producing that can help plants deal with various forms of stress. Moving on to the last page, nutrition, we can see the format-wise, everything pretty much the same as previously. Of course, we have carbon included as a major compound with N, P, and K given its relevance to soil health. And in this particular report, we can see in terms of the PGRs and stress adaptation metabolites, things looked pretty good overall. Here in the nutrition pathways, the carbon over on the left, we have our overall pathway, and then on the right, we have our specifics. And overall, carbon and nitrogen look pretty solid. Carbon particularly high, nitrogen in that medium range with some room for improvement, but phosphorus and potassium in the lower range, the area that may potentially be improved upon with biological products or soil management practices that can improve these areas of soil health and increase the levels of microbes that can unlock, solubilize, and cycle phosphorus and potassium. And then lastly, our 8 micronutrient compounds, the same pathways from our previous reports, and in this case, they're all within pretty optimal ranges aside from chlorine in the lower range. So that very, very quick overview of our new BeCrop report format, and of course, things generally look pretty much the same other than the soil quality and resilience metrics in our soil quality section. But if you do run into any issues interpreting or better understanding the updates to the reports, feel free to reach out to us or shout out a question now in the chat. And without further ado, we'll move on to questions and go ahead and address any that we might have. Thank you.
Thank you so much, Gus. That was great. We have a lot of questions that have come in, so we're trying to, we'll try to get to as many as we possibly can and then reply to the rest of you guys via email. First question that came in, how does BeCrop align to support the 7 principles of soil health?
7 principles of soil health, meaning like protection, looking, I mean, I think it's thrown around. Sometimes it's 7, sometimes it's fewer, sometimes maybe it's more, but I could probably spend an hour going into detail just on that, but I'll just touch on some of the big ones that many of us are very familiar with. So one of the really overarching ones is ensuring protection of that soil. When we have bare ground and fallow fields, of course, the soil microbiome is subject to a lot more stress because the sun beating down on it, no plant roots there to prevent erosion. So in that case, of course, tightening rotations and cover crops are really the key there to offer that protection to the soil and ensure that there are roots in the ground as well as above ground biomass to help modulate those effects of weather and erosion. And there's definitely differences in terms of what cover crops offer. So of course, legumes offer, nitrogen fixation. We have other cover crops that improve soil texture, can improve penetration of the soil and water penetration. In fact, right behind me here, I have the USDA's cover crop chart, which just provides a rundown on many of the different cover crops used in the US. And with BeCrop, what you can looking at soil microbiome functionality and all the metrics we have are many of them can inform what specific cover crops you advise or recommend to your clients. So looking at, if levels of if you're looking at carbon fixation or nitrogen, you might look at crops that you might try to incorporate grasses, a mix of grasses and legumes that can help offer the nitrogen fixation to provide nitrogen from the air. And then as that cover crop is chopped and dropped or roller crimped, you can improve the levels of organic carbon and thus potentially contribute to the levels of carbon nitrogen mineralizing microbes. Similar with carbon, you can look into specific cover crops that may help promote higher levels of carbon biomass in the soil in the form of the organic matter decomposition they offer. So that's just one example there. But as far as principles of soil health, we've really designed a lot of our BeCrop report kind of revolves around the areas in which management can promote soil health. Another example is with our improved, with our new resilience metric. That's looking at basically that can inform the risk of tillage, fungicides, other types of management practices that we know can be somewhat disturbing and have negative effects on the soil microbiome. So higher resilience would indicate less risk, and lower resilience would indicate that fungicides, tillage, other forms of disturbance can have more of a negative detriment on the soil health. So those are just a couple examples. I could go on for a while, but I'll save some time for a few other questions, but really happy to chat offline or follow up with whoever asked that question to get more specific.
Amazing, Gus, thank you. There's a question here and another one. I'll combine the two. One is about how do we differ from other companies that offer biological soil testing? And another person asked that I noticed your sequencing is limited to 16S and ITS. Are we thinking about expanding this to include Illumina shotgun sequencing and so on? So your thoughts?
Yeah, great. That's a great question we get all the time. So in terms of the type of sequencing we use at Biomakers, we do stick to 16S and ITS amplicon sequencing to identify and quantify the bacteria and fungi in the soil. Now that of course provides, the species that we're able to see because 16S and ITS are very robust and reliable indicators of species. And 16S and ITS are also very effective at detecting a wide breadth, a wide scope of microbes in the soil. So, but the way that we calculate functionality goes beyond that to basically a second step in the process, and that is where our database comes in. So Biomaker's microbial function database looks across peer-reviewed research and looking at genes, so looking across research that has taken into account metagenomics, full genomic sequencing, to tell us what those microbes do in the soil and how do they interact ecologically. So really, we rely on 16S and ITS because it's a very effective indicator of identifying species. But then the real strength in the tangible data we provide, the way that we do that is through our database, in which we take those species we've detected in a sample, we run it through our database to determine these functional metrics that are on the report. So, you know, metagenomics and full genomic sequencing, of course, shotgun sequencing, whatever you want to call it, does look across not only the species, but their full genome to see what genes they possess. But with our technology, we're able to take, you know, the species that are there and then infer functionality based on a wide breadth of data. So we're our goal is really to take sort of the best of both worlds, and that's why we've gone with 16S and ITS sequencing over what some of our competitors have done with shotgun sequencing.
Great, thanks, Gus. Next question is, how do we relate BeCrop results with a chemical soil analysis result?
Great question. So I think it's very useful to treat the BeCrop like a complementary test to chemical fertility analysis. So taking, you know, a Mehlich 3, whatever, you know, typical chemical fertility analysis you run, and then looking at it side by side with the BeCrop, which we also offer the chemical fertility with our BeCrop Plus as an optional add-on with our partnership labs like Waters and Regen. So if you look at those reports side by side, looking at the BeCrop and the chemical fertility, you're kind of looking at two aspects of fertility: what's there in the soil and plant available in terms of the chemical fertility tests, and then the biological fertility, what microbes are there that can unlock and promote plant uptake. And if you're looking at them side by side and you see that you have, you know, what a lot of growers run into is they've been putting out, you know, their fertility program's been increasing for years, but they're not getting a yield response. And if they are looking at just chemical fertility, oftentimes they'll see that they have optimal levels of nutrients in the soil, you know, fairly well balanced, but yet they're not getting those nutrients into the plants, and it's limiting their yields and causing, they're noticing issues with nutrient deficiencies in their tissue analysis. So then if they take the BeCrop test and look at that complementary with the chemical fertility test, they might see that, you know, phosphorus and potassium, for example, might be, you know, rated low. Specific nutrients that they might be seeing deficiencies in that measured high in abundance in the chemical fertility test, the biological cycling in the BeCrop report may not be optimal. And in that case, what this is pointing to or saying is that the issue, the bottleneck they're running into, is more likely a biological issue than a chemical fertility issue. On the flip side, if you run a BeCrop and a chemical fertility test side by side, you might say, look, the biology looks great, you know, nutrient cycling looks optimal, you know, carbon, nitrogen, phosphorus, potassium, the micronutrients all tend to be in the medium to high range, but maybe on that chemical fertility report, they see low levels of specific nutrients or imbalances in ratios between certain nutrients. So really, what we do by offering BeCrop along with chemical fertility is provide a way to look at the whole picture, not just the biological fertility, not just the chemical fertility, but by looking at them together, it just makes it a lot easier to identify what is deficient, what can be improved upon, so that we're not continuing to do a management practice or you know, purchase inputs that aren't directly addressing the issue. That's a great question.
Absolutely. The next question we've got is fungi bacteria ratio based on species count or biomass? Have you done any work looking at correlation of FB ratio with BeCrop versus PLFA or microscope analysis? Is it realistic to ever get to 1-to-1 on BeCrop test?
That is a very good question. That's something we're very interested in looking in. It's important to note that the fungi to bacteria ratio on the BeCrop report is very different from PLFA or microscopy. So microscopy, PLFA, and genomic sequencing are all really basically different lenses of looking at the same thing, but they're vastly different because, of course, when we look at DNA, that's on a whole different level than looking at phospholipid fatty acids in the case of PLFA tests, or looking through a microscope. Each technique is, of course, going to have some positives and negatives, some trade-offs in terms of how representative that fungi to bacteria ratio is. For example, in PLFA, there's definitely undifferentiated phospholipid fatty acids on reports that aren't accounted for. And what I will say is, so I would exercise caution in comparing these. As we continue to increase our databases and get more data on soil samples that have had the BeCrop conducted and PLFA conducted on them, and maybe even microscopy, we'd like to look and see how those correlations work out. But to answer the first part of that question, when looking at fungi to bacteria ratio, we're not just looking at species, we're looking at the DNA representative of total biomass. So we're not just looking at number of species, but also taking into account their abundance, because otherwise you might have a lot of different fungal species, but they're present in really low levels. And that would be a little bit of a misconception of interpretation if fungi to bacteria ratio looked high just because there were more fungal species. So that is how we look at it. And as we continue to get more data, I'm interested to see how our fungi to bacteria ratio correlates with PLFA and microscopy. But for now, I would say it's kind of an apples to oranges comparison, and I would stick to comparing within BeCrop reports, looking at changes in fungi to bacteria ratio, or within PLFA. And maybe if you have both on hand, look at similarities and differences, but not compare one PLFA to one BeCrop.
That makes sense. Next question: will adding a growth hormone product be more or less beneficial with soil expressing natural high levels of hormones producing bacteria? What about low levels?
That is a great question, and this is an area that we're really interested in, and again, getting more evidence to really support our hypotheses. I would say right now the working hypothesis, and again, this is based on anecdotal evidence and sort of theoretical evidence, is that within the soil, so it's important to keep in mind that the plants and microbes don't just have a one-way interaction. It's a two-way interaction where as the plant continues to grow and metabolize, it sends out root exudates, which are basically sugars that can contain other signaling molecules into the soil. So those root exudates basically feed the microbiome, and they also signal to the microbiome what the plant needs. And when a plant does need certain growth hormones or metabolites, and it signals to the microbes, it can stimulate those microbes that can provide those growth hormones, those phytohormones or plant growth regulators in the soil. Now, if a plant is getting PGRs applied through foliar methods, it may not put out those signals into the soil to request that the microbes produce and supply those PGRs. And this is very similar to what we have seen in some evidence with micronutrient foliar applications. So if a plant is if the PGRs or foliars being applied above ground to the plant to the foliar biomass, then the plant isn't going to communicate the same with the microbes and encourage them to produce those PGRs. This is a working hypothesis, so as we get more evidence and maybe we'll have a research project come around that looks at this and publishes the results, we'd be very happy to share that in our tech validation area on our website. But for now, that's just the working hypothesis. And if anyone is interested or has a client that maybe sells PGRs or is looking at this is definitely something that we can evaluate with BeCrop and determine if there is a negative effect of PGRs on natural production of PGRs in the soil. Great question.
Okay, we have time for maybe a couple of more. The next question is about our AI. They're wondering if you can give a little bit more information about our AI process. What does the AI in particular influence a trial?
So I don't think I should go too deep into specifics here, but I can provide kind of just an overview of how our machine learning and AI really improves upon our data overall. As we get more samples in our database and improve the number of taxonomic references as well as the number of soil microbes we've detected, and then increase the number of samples looking at how those microbes interact, our AI can take that information as feedback that improves the overall robustness of our algorithm and how we calculate these functions. So that is something that has been updated and continues to be updated with each new iteration of BeCrop. I don't know, within, data science may be able to provide some deeper in-depth knowledge on how that machine learning works, but I know that for the protection of our intellectual property, I should probably kind of stick to the more general details. But that's essentially how our machine learning and AI works. Is it's an iterative process that provides feedback based on higher improvements and higher numbers of samples in our database. That ultimately feeds into the algorithm and improves the accuracy and robustness of our results over time.
Thanks, guys. We have a couple of questions about the Haney test, and a couple of people are wondering if it could be integrated with the BeCrop analysis to enhance the overall usefulness of both data sets.
Yes, that's a great question. Something that we've already begun to do in our partnership with Regen Ag Lab. With samples sent to Regen or sent straight to our lab at Biomakers, we can offer the Haney test in conjunction and subsample take a soil sample that's been sent in, break apart a subsample, and then have Regen analyze it, or vice versa. A sample sent to Regen can be subsampled and then forwarded to our lab to run the BeCrop test. So the Haney and BeCrop can be run in conjunction on one sample, and we have integrated the results into what we call our BeCrop Plus report, which will include the Haney data on the last page. Now we're continuing to improve this process because the Haney, of course, is a very in-depth report and a lot of metrics on there. Currently, we don't include all of the metrics on our BeCrop Plus report, but the full Haney report from Regen Ag Lab is sent out with each BeCrop result. And I'm a big fan of the Haney test. I think that it's a great test for looking at chemical fertility through a very soil health focused lens. And I think in particular, there's a lot of relevance in correlating patterns and trends between the biology in the soil and what the Haney test can look at. And particularly, the Haney looks at carbon very closely and also looks at different forms of both nitrogen and phosphorus, both organic and inorganic forms. What we look at in the BeCrop are the microbes that drive the changes in those nutrients between the organic and the inorganic form. Use the carbon that we look at in the Haney test to drive their metabolism and support overall microbial functionality. So I think the Haney test is an incredibly good test to use in conjunction with BeCrop to look at both the chemical fertility, the biology, and then we can even look at, in terms of nutrient cycling, how some of the biology and what we're seeing in the Haney test are influencing each other. So yeah, I think that as moving forward, there will be a lot of really cool research that comes out with the Haney test, kind of looking at what we measure in the BeCrop and how that functionality carries over into nutrient availability and plant uptake.
Great, and for everyone on the webinar, we are actually planning on organizing a webinar sometime in May with Regen Ag Lab to talk about Haney test and BeCrop technology. So be on the lookout for that webinar. We'll just take one last question.
I just wanted to mention, we did do a webinar a few months back, touching base with Regen Ag Lab and introducing our partnership. So that webinar on our website may also provide some additional background info on the Haney test and how it can integrate with BeCrop.
Absolutely. One last question we'll take before we end this webinar. The question is about our portal, and they're asking if multiple people will have access to the dashboard, which would include the client, consultant, or research manager, grower.
So that's something that we can already offer. So within each BeCrop account, we have the option through request. So if requested, we can create sub-client accounts so that, say that you have multiple clients and you want them to be able to log in, but only to view their results. Of course, you don't want all of your clients able to see all the results across clients, because in often cases, we want to be able to divvy up the results to whatever client they're for. And so that's something that we can do. We can, by request, add sub-client accounts, and then you can decide what reports and data to assign for that account to be able to see. So yes, that's something that we can offer. Very important for managing data and ensuring that there's not any improper sharing of information.
Great. Well, thank you, Gus, for giving us your time today and providing a great overview of BeCrop 3.0. Thank you, everyone, for joining the webinar today. We hope that it was useful for all of you. As we mentioned earlier, a recording of this webinar will be shared with you, along with information on BeCrop Advisory Program, and if you're interested in joining. And of course, we have tons of webinars on our website if you want to go ahead and take a look at those and watch them at your own time, and we'll definitely keep you posted regarding future webinars, especially one with Region Ag Lab. So thank you all again for joining, and we all hope you have a wonderful rest of your day.